Hello everyone, I hope you're doing well. My name is Dianiris Astacio. I'm a third-year biomedical sciences student, and today I'll be giving you a brief presentation about the effects of stress and exercise on CRH levels in a Sprague Dawley rat model of endometriosis. The aims of this project are to study the change of CRH levels in the brains of rats with endometriosis exposed to exercise and observe the effects of the activation of the HPA axis on endometriotic lesions in Sprague Dawley rats. As seen on this diagram, the HPA and HPG axis have an effect on the female reproductive system, but we will go into details later. We hypothesize that exercise alleviates endometrial lesions by the inhibition of central gonadotropin-releasing hormone via CRH mechanisms. Before continuing, I'm going to briefly discuss some known facts of endometriosis. It is known that 1 out of 10 women of reproductive age have endometriosis, which is characterized by a growth of endometrial tissue in areas outside of the uterus. Symptoms include painful cramps, longer menstrual periods, heavy flow, vomiting, bowel disorders, and infertility, making this a debilitating and very serious condition. The diagnosis can only be made via surgery and few treatments are available, but they are mainly hormonal. For example, Lupron. Lupron works by cutting gonadotropin-releasing hormone secretion by a negative feedback mechanism, as shown in this diagram. This inhibits the production of sex hormones that can exacerbate the effects of endometriosis. It is known that environmental factors, such as stress, can worsen endometriosis by a direct impact on the HPA axis. HPA stands for hypothalamic pituitary adrenal axis, which is a neuroendocrine system that controls glucocorticoid production and secretion. Although it can be voluntarily controlled, it can be influenced by environmental factors, such as stress. With stress, the hypothalamus secretes corticotropin-releasing hormone, or CRH, which in turn stimulates adrenocorticotropic hormone, or ACTH release, as you can see here. ACTH is a tropic hormone that promotes cortisol production to mediate a stress response in the body. The HPA axis is controlled by a negative feedback. Cortisol inhibits CRH and ACTH release, ending the stress response. We are interested in the role of CRH in this response. As discussed earlier, CRH is a neuropeptide that stimulates the production of ACTH. It acts in the pituitary and participates directly in the stress response. Here we can see its structure. CRH can mediate this response via 2 receptors, CRH receptor 1 and CRH receptor 2. Receptor 1 is known for having a role in pro-inflammatory mechanisms, while receptor 2 plays a role in the anti-inflammatory response. Both of these receptors have a G-coupled protein structure, shown in these pictures. Now, where can we locate them throughout the body? Here, we can see a distribution of the CRH receptors 1 and 2 all over the body, but we are focusing on the brain and reproductive system, which are the tables you see to the left of each body. Here, you can see the expression of the receptors on the mentioned tissues. CRH receptor 1 is mostly expressed in the cerebellum, amygdala, and endometrium. CRH receptor 2 is highly expressed in the cerebral cortex and has relatively high expression in the fallopian tubes. Now that we know the location of the CRH receptors, we must discuss what is known about CRH in endometriosis. Research shows that CRH and its receptors are significantly expressed in endometrium outside the uterus, helping maintain inflammation and local stress. Also, it has been shown that CRH was decreased in the hippocampus and increased in the ventral paraventricular hypothalamus in animals under stress with endometriosis, suggesting the activation of a stress network. CRH can maintain inflammation through galectin-1, Why? Because CRH can upregulate in the CRH receptor 1 dependent matter endometrium galectin 1 expression, which in turn stimulates cell proliferation in endometriotic lesions. In addition to this, it is known that antilarmin, a CRH receptor 1 antagonist, can reduce endometriosis vesicles in size and amount. We have this information, and yet the role of CRH and the receptors has not been well understood with endometriosis and exercise. Exercise is known for its beneficial effects in endometriosis and in many other conditions. To understand more about this, let's look at the diagram on the right. The HPA axis has a direct relation with the hypothalamic pituitary gonadal, or HPG, axis. CRH and glucocorticoids from the HPA suppress gonadotropin-releasing hormone from the HPG. The stress of exercise activates the HPA axis, which produces CRH and in consequence inhibits gonadotropin-releasing hormone. It is known that an inhibition of the gonadotropin-releasing hormone can alleviate endometriosis lesions and symptoms. Here's a good time to remember what we talked earlier about the mechanism of action of lupron that works by inhibiting gonadotropin-releasing hormone by a negative feedback inhibition. Another factor to consider is that during exercise, beta endorphins are secreted. Beta endorphins are the natural painkillers of the body, and they can act on delta opioid receptors of gonadotropin-releasing hormone neurons to cause hyperpolarization and inhibit secretion of gonadotropin-releasing hormone. This effect is fairly specific because delta opioid receptors are only expressed in mature gonadotropin-releasing hormone neurons. Stress and exercise can inhibit gonadotropin-releasing hormone, but mechanisms and their role in endometriosis are still unknown. To prove our hypothesis, we use the rat model timeline presented in this diagram. The rats were subjected to surgery for the endometriotic implants, and 7 days later they started to exercise. 2 weeks after the surgery, rats were exposed to stress for 6 days. On day 21, open field and elevated mace tests were made to measure the rat's anxiety. These tests were repeated on day 59, and on day 60, the rats were sacrificed. For visualizing CRH expression in the hippocampus of the rats, we use staining techniques such as immunofluorescence, as seen on the left, and immunohistochemistry, as seen on the right. After this, we expect to find reduced lesions on rats exposed to stress and exercise with endometriosis, larger lesions in rats exposed only to stress, and significantly lower lesions on rats that were only exercising. In addition, in the brain, we expect to see reduced levels of CRH on rats exposed to stress and exercise with endometriosis, higher levels of CRH in rats exposed only to stress, and lower levels of CRH on rats that were only exercising. This would explain, or at least in part, the impact and importance of exercise for endometriosis patients as an alternative treatment. With this, I conclude my presentation. I would love to thank Dr. Gladys Chompré for the guidance and mentoring during this process, Mireya Cruz, Dariangeli Pacheco Cruz, my partner, and Dr. Caroline Appleyard for allowing us to be part of this project and the Appleyard students that support the lab. To finish, here are the references. Thank you for your attention. Have a lovely day.